good morning or good evening, everybody. So depending on where you are, my name is Samie Jaffrey. I'm at Cornell Medical School in New York City and one of the principal investigators of the Center for Genomic Information Encoded by RNA Modifications. I'm here along with Kate Meyer, another lead principal investigator on this center, which is funded by the NIH and the National Institutes of Human Genome Research. And one of the things that the center and the NIH are interested in doing is helping to develop or encouraging the development of new methods and technologies that can be used to study RNA modifications. And in particular, we're really interested in moving away from sort of the qualitative analysis of modifications where people could see that, okay, modifications here or not here, but getting down to very quantitative measurements of RNA modifications with, exact stoichiometries of modifications, exact localizations, and really allowing us to start to study questions about dynamics and whether or not these modifications are changing in different contexts. And the big problem has been the methods, and the methods methods that, Kate and I, are responsible for some methods which, I think have done some good in terms of help identifying the importance of M6A throughout the transcriptome, but have also been problematic because I think a lot of people use those methods to infer quantitative changes in M6A, which don't necessarily exist. And so the point of this symposium is to really explore some of the newest methods, as well as some of the science that's come from these methods. But we're really interested in the methods and the technologies and the latest innovations. And what we want to do is we want to come up with ways that researchers can make decisions about what are the best methods that can be used for their research. So we really want to democratize modification analysis. We want to make it easy for everybody. Kate can probably tell us a little bit about the different types of talks that we're going to hear today. And then I'll talk a little bit about the ground rules and how everything is going to be conducted, and how everybody can ask questions and participate. But Kate, maybe you can give us just an overview of the different things that are going to happen today and tomorrow and the point of the panel sessions.
Sure, yeah. So again, welcome everybody. We're excited for the next couple of days, and hopefully people have had a chance to take a look at the agenda. We've got a fantastic lineup of speakers happening today and tomorrow, and I think hopefully some really interesting and informative panels sessions that we have planned. So today for day one, we're going to be spending the morning talking about mRNA modifications with a particular focus on M6A. So we'll have several talks, including mine, which is going to be the first one in a few minutes here, talking about some recent developments in terms of methodologies for detecting and quantifying M6A. And then we'll have after that a panel session with all the speakers where, as Samie kind of mentioned, the idea is we really want to try and, have a good discussion and come to some, conclusions if we can about best methodologies to use. And so like I said, this morning we'll be doing that for M6A. Then we'll have a brief lunch break, and then in the afternoon we're going to have some talks focusing on new instrumentation for quantifying RNA modifications, and that will be followed up with a panel for basically how to select and use some of these newer methodologies that have come online, including things like Oxford nanopore technologies for mapping modifications. So that'll be today, and then tomorrow, day two, we'll start off the morning with several exciting talks focused mostly on methods for quantifying and mapping modified nucleotides and tRNAs. That'll be followed up by a panel where, again, we're going to try and focus on these methods and what's the right method to use and when, what are the pros and cons. And then tomorrow afternoon, we're going to have a similar speaker followed by panel session focusing on the mapping and quantification of ribosomal RNA modifications, and a panel session focusing on some of the major challenges for rRNA modification detection. And then at the end of the day, on day two, we're going to wrap up with one final speaker session where we're going to hear some really exciting emerging methods for installing and manipulating RNA modifications in cellular RNAs. So that's basically what we've got planned for the next couple of days. We're very excited. We're happy to have everyone here and hoping we'll get some good discussions. And we've got a great lineup of speakers, so we're looking forward to it.
Sure. All right. So with that, I'm looking forward to an exciting meeting for the next 2 days, and hopefully people will get a lot of their questions answered. So with that, I'm going to hand the microphone over to Kate Meyer from Duke University, who's going to present on transcriptome-wide profiling of M6A in single cell.
All right, thanks very much. So like we just said, we're starting our day off here with discussions of methods for mapping and quantifying M6A. So I'm really happy to tell you about some of my lab's latest work in this area. So I think a lot of people probably in this meeting are no stranger to M6A, but this is certainly not everyone's area of expertise. So by way of just a very brief background, M6A is the most abundant internal mRNA modification. It's found in thousands of cellular mRNAs. The vast majority of M6A sites are deposited by a single methyltransferase called METL3, which interacts with other key proteins like METL14, WTAP, and a handful of others to target RNAs for methylation. And so this is a process that occurs co-transcriptionally immediately as the RNA is being transcribed. And M6A is deposited in a unique consensus motif, which is most frequently a so-called Dratch motif, but really at its core is basically a purine AC motif. And despite the relative high frequency of these motifs throughout a given mRNA sequence, we see this very unique distribution of M6A in mRNAs, which is characterized by enrichment in long internal exons, as well as at the beginning of 3' UTRs and kind of in the vicinity of the stop codon. And some very recent exciting discoveries revealed that this is largely due to occlusion of these Dratch motifs by exon junction complex proteins, which helps gives rise to this unique distribution of M6A. So in terms of M6A function, really at this point, M6A has been implicated in virtually every aspect of the mRNA life cycle, but some of its most consistent links have been with promoting mRNA decay. And M6A really has these unique functions by regulating RNA protein interactions. So that can be either indirectly through controlling M6A structure and then the availability of RNA binding motifs by RBPs, as well as directly by things like recruiting M6A reader proteins. And so a very important class of M6A readers that I want to highlight are the YTH domain containing proteins. So these are 5 proteins that exist in humans, all of which have this highly conserved YTH domain, which is essentially just an M6A binding domain. And so be it through these YTH domain containing proteins or other reader proteins or other influences on RNA protein interactions, like I said, M6A has a variety of different functions within the cell. And so Samie kind of alluded to, some of the first methods for discovering M6A transcriptome-wide. Those were developed in his lab when I was working as a postdoc, as well as by Gideon Rakavi's group. And so these are all antibody-based approaches called MeRIP-seq or M6A-seq, which is very simply immunoprecipitate methylated RNAs using these M6A-specific antibodies. These have by far been the dominant tool that have been used to map M6A transcriptome-wide and are still widely used today. Although, as Samie mentioned, they're not without their limitations. And one of the most difficult limitations, at least for my group, which often finds itself trying to map M6A from very small amounts of input material, is this requirement for a lot of RNA before you do this immunoprecipitation reaction. And so this has really limited studies of M6A in things like distinct cell types or subcellular regions, or in precious samples that might not be, very abundant, like human samples or patient tissue. These limitations are certainly things that the field has recognized, and in the past several years, there have been several new antibody-independent methods that have been developed, some of which we're going to be hearing about later this morning, which will be very exciting. But nonetheless, a lot of these methods still require, a decent amount of RNA to do the mapping. And so there's been this big challenge of, moving M6A mapping transcriptome-wide from bulk cells, which is what the vast majority of studies have done, to single cells. And so why do we need to do this, right? Why do we care about M6A in single cells? Well, we really don't know much about the content of M6A in different cell types that make up unique tissues. So my lab in particular is very interested in the brain, and the brain we know is made up of all kinds of different cell types, right? But even though there have been many MeRIP-seq and other, antibody-based brain profiling M6A data sets that have been put out there and published, we really have a very poor understanding of how methylation might differ within these complex cell types that make up the brain. And this is true of virtually every tissue too. Furthermore, we study, Samie talked a lot about M6A quantification, and as a field, we care a lot about M6A stoichiometry, but really all of the measurements of M6A stoichiometry for the most part that we have are from, bulk cells. So if we think of an M6A site that might be, in a given mRNA at 25% stoichiometry, well, what does that really mean, right? Does that mean that, all the cells in that population have 25% M6A at that site, or that, a quarter of the cells have 100% M6A at that site, or somewhere along that spectrum? We really don't know. Similarly, even the early M6A mapping studies have reported that M6A sites can cluster within mRNAs, but until we can map M6A in individual cells, we really don't have a good understanding of how frequent individual sites within a given RNA might be throughout different cells of the population. Are they all there in all the cells, or are they there at different frequencies? And does this even matter, right? But until we can answer this, it's really hard to know. So for all these reasons, it's really important, we feel, to be able to map M6A in individual cells. And so to do this, we realized that we need to develop new methods. And so we developed this method called DART-seq, which stands for deamination adjacent to RNA modification targets, which works by installing a unique mutation signature adjacent to every M6A site. And so the way that we achieve this is we take the YTH domain of YTHDF2, which is a very important M6A reader protein in cells. You remember, this is the domain that's basically an M6A binding domain. So we take this M6A binding domain, fuse it to a protein called ApoBec1, which is a CDU deaminase that normally targets a small number of RNAs in the cell for CDU deamination. So the idea then is that every time this protein contacts an M6A site, it gets recruited there by the YTH domain, and then conveniently, because M6A is always in an RAC motif, there's always a cytidine available in the vicinity for CDU deamination by the ApoBec component of this fusion protein. So now all we need to do is mix this with RNA, and we can do that either in vitro by purifying this protein, or more frequently what we do is express it in cells, let the enzyme bind to M6A and do its editing, and then collect RNA. And this is basically just now an RNA-seq experiment. We can map these CDU mutation events and infer the location of M6A. And so we published this back in 2019, and basically showed through a variety of different methods or a variety of different ways that I won't have time to take you through today that it works. And we showed that we could use this technique to map M6A in the equivalent of about 1,000 cells, which is a pretty good improvement. But this wasn't yet single cell, right? And so we really wanted to push for single cell mapping. And so this was all work that was spearheaded by this super talented postdoc in the lab, Matt Tagawski. And so the first thing that Matt did was he generated a stable HEC293T cell line that had inducible expression of our ApoBec YTH enzyme. And he also made a second cell line that had a mutant version of this enzyme. So in this mutant version, we deleted part of the M6A binding domain of the YTH domain. And what this led to was very, very severely compromised M6A binding. So we look here at an RNA pull down followed by a Western blot. We can see that our mutant protein has a very diminished ability to bind methylated RNA compared to the normal protein. So now, because of this unique design of the induction system, Matt could use a combination of fluorescent sorting to sort individual cells and use plate-based SmartSeq2 single-cell cDNA synthesis technology, as well as he used the 10x Genomics platform, which enables partitioning of individual cells through microfluidic means and enables sequencing from a much greater number of cells. So we kind of combine these two methods, and collectively we sequenced M6A in over 11,000 individual cells. And so to map these M6A sites, we kind of modified this bullseye pipeline that we had developed, and that was developed by a former postdoc in the lab, Matthew Flaman, to identify these M6A sites in cells based on the CDU mutation signature. And so when Matt did this and looked transcriptome-wide, he saw basically that it worked. So if we look at all of the cells as a population, we can see that the CDU mutations in the cells that express ApoBec-YTH match the distribution of M6A, shown here in red, whereas the cells that have expression of our mutant protein, which is M6A binding deficient, shown in blue, have a different distribution. He could also do this for just randomly selected individual cells throughout the population. So this distribution of M6A holds within individual cells. We could also just do some other kind of QC, for instance, looking at the motifs that these CDU mutations are enriched in. We see that they match the M6A motif. We can also show that the RNAs that Matt identified in single cells overlap well with RNAs identified in the same cell type through antibody-based or bulk DART-seq-based approaches. And so one of the first things that really stood out to us was just the sheer number of sites that we identified. So we found over 170,000 M6A sites in just shy of 10,000 RNAs. And this was really many, many more sites than bulk studies had previously been reporting. So we dug a little deeper to this and we looked in individual RNAs. And so we compared HEC293 T cells where we did bulk DART-seq compared to single cell DART-seq. And we saw that in the bulk cells, which matched, antibody-based studies, most RNAs had just a couple of sites, and there were very few transcripts that had, lots of hypermethylation. Whereas in the single cell data set, most mRNAs had, a dozen or more M6A sites, and there were hundreds of transcripts that had, a lot of sites, over 50 sites. However, when we looked at the distribution of these sites within the population, we saw that they're relatively rare. So the vast majority of sites are found in fewer than 20% of cells in the population. And so just another way to kind of illustrate this is by showing an individual transcript that's representative of the vast majority of the transcripts that we looked at. So this PPP2CA RNA, what we're looking at here is the gene architecture on the bottom with introns, exons, and 3' UTR, 5' UTR. And then we have a bar here that represents every site that's been identified in MET single cell data. So this RNA has 37 total sites. And the height of these bars indicates the proportion of cells in the population that have methylation at that site. So hopefully what you can appreciate is that the majority of these individual sites are found in, a relatively low proportion of cells in the population. And even at its highest, there's only a couple of sites that are still found in fewer than 40% of cells in the population. And interestingly, when we look at the number of sites in this RNA in cells that express the RNA, in any given cell, there's usually only 2 or 3 individual M6A sites. So this was all really interesting because this is, again, novel stuff that was unexpected that we hadn't previously appreciated from bulk M6A mapping. So the next thing we wanted to investigate was M6A stoichiometry. And we kind of, can revisit this question of what does 25% M6A mean in a population at a given site. And so when we first published DART-seq, we had shown with synthetic RNAs that the amount of CDU editing adjacent to an M6A site was proportional to the amount of M6A. And so to see if this was the case in Matt's single cell data, he compared his percent CDU values from his single cell data sets to 3 individual methods for M6A mapping and quantification that have been developed. So Maze-F cutting or master-seq, Scarlet, and an RT-qPCR-based approach. And in each case, he saw a positive correlation between the amount of M6A and the proportion of CDU editing. So I want to be very clear because we're not yet at the point where we can equate a given percent CDU to a given percent M6A, but we can use it as a proxy. And what that does is that lets us kind of make some, hypotheses regarding M6A stoichiometry in single cells. So with that knowledge, we then, looked at M6A stoichiometry, and we wanted to kind of compare what we see in single cells to what we saw with bulk sequencing. So basically, when we looked at the distribution of percent CDU values in both of our single cell DART-seq data sets compared to bulk mapping from the same cells, we saw that there was many more high abundance M6A sites and a much broader range of stoichiometries in the single cell data compared to the bulk data. And what we looked at when we looked at this more kind of transcriptome-wide and individual RNAs is that the abundance of M6A at any given site, stoichiometry, is really highly variable across individual cells. And so I can illustrate that probably best by showing you an example transcript. So this is an RNA that's expressed in the vast majority of cells in the population. And what we're looking at is methylation of an individual adenosine within the 3' UTR of this transcript. And so this is just a UMET plot, which basically clusters the cells in the population that express this RNA. And now we've colored each cell based on the proportion of CDU, right? So the amount of M6A. And hopefully what you can appreciate by looking at this UMAP plot, first of all, is that there is a lot of heterogeneity in the amount of M6A at this site. And when we look at that just as a histogram, we can see that if we took the population average percent CDU value, which is equivalent to, bulk data that we've had in the field, right? We see that hovers around 20%, but that doesn't really tell the full story, right? Because there are many cells in the population that actually have no methylation at that site. And additionally, there are many cells that have much higher levels of methylation at that site. So really, I think what this tells us is that, these population level estimates of M6A stoichiometry fail to detect the extremes of abundance of M6A within single cells, which can be highly variable. So these are some of the major conclusions that we drew. We published this last year. We did some other really interesting analyses. So if you're interested, I urge you to check out the paper. But what we've been working on since then is trying to move toward more physiologically relevant systems. So what we did to that end is we've generated DART mice. So these are transgenic mice that express the ApoBec YTH transgene. So we designed these mice so that it's an inducible lock-stop-locks, so pre-dependent system, where basically we can inject the mice with tamoxifen, and that will lead to excision of a stop codon that enables translation of the ApoBec YTH transgene. And these are under the control of a ubiquitous driver line. So when we looked at, just to confirm that the expression of this transgene could be induced, we looked across a variety of different tissues and saw good induction. We also made the ApoBec YTH mutant version of these mice that we use as a control for background editing. We could also just kind of spot check, look at individual RNAs at M6A sites in these different tissues and see that there was CDU editing adjacent to these M6A sites only in the ApoBec YTH mice, not in the mutant mice, which was very reassuring. So then we said, okay, let's see just in bulk tissues first. Let's see if these mice can be used to map M6A transcriptome wide. So again, given our lab's interest in the brain, we started with the brain. We isolated 3 major brain regions, cortex, cerebellum, and hippocampus. Saw good induction in each of the regions. And then we performed CDU or we performed RNA-seq and CDU mapping using Bullseye to see where the M6A sites were. This revealed thousands of sites across the 3 different brain regions. Reassuringly, these sites were enriched in the general vicinity of where M6A is usually found, including in the vicinity of the stop codon, as well as within long internal exons. And we could also look at the RNAs identified in these mice by DART-seq mapping and compare that to data sets in the same brain regions that have previously been published using antibody-based methods, and we saw a high degree of overlap. So this was really good because basically it told us that we can indeed use these mice to map M6A in vivo. We were really curious to see how much unique versus common methylation there was in these brain regions. Previous studies have looked at this to some degree and reported that most mRNAs are commonly methylated, but there is some unique methylation. And that's pretty much what we found. So even if we control for RNA abundance across the 3 brain regions, we see that the vast majority of RNAs are methylated in most brain regions, but there is some unique methylation that occurs that might be brain region specific. So we're currently following up on that. So like I said, this was great. It tells us these mice can be used for mapping M6A in vivo, but now can we use them to map M6A in single cells? That was our original goal. So we did just that. We focused on the mouse cortex. We saw good representation of the major cell types that make up the brain, similar to what we found in HEC293 T cells. When we mapped the M6A sites, we found hundreds of thousands of M6A sites. We looked at the distribution of these sites and saw that, again, they matched the distribution of M6A, both at kind of the bulk level when we combine all the data sets together, or all the cells together, as well as within individual cell types. We see the same general distribution of M6A. And interestingly, when we looked at the number of sites in each of these different cell types, we saw that most cell types have very similar numbers of M6A sites, but microglia really stood out to us as having many fewer sites compared to these other cell types. The first thing we thought was, well, surely the ApoBec YTH transgene is somehow just not being turned on as well in microglia compared to the other cell types, but that didn't turn out to be the case. So at the RNA level, everything was very similar, and at the protein level, if we isolated microglia and compared to astrocytes, which is a cell type that had very similar levels of M6A sites to the other cell types in our data set, we saw no substantial differences in expression of the ApoBec YTH transgene, shown here by an HA stain. We We also performed immunofluorescence in microglia and astrocytes in brain slices isolated from the transgenic DART mice. And again, we saw no significant difference in protein expression using immunofluorescence. And then importantly, we could actually validate this. So we isolated microglial and astrocyte mRNA from wild-type mice and subjected it to mass spectrometry and confirmed that there is indeed a substantially reduced amount of methylation in microglia versus astrocytes. So this was a really unexpected discovery that we're excited to follow up on in the lab. So another thing that we were really interested in was looking at differential methylation across cell types. To do that, we basically computed the average percent CDU values of all the M6A sites identified in each individual RNA in the data set, and then just did pairwise comparisons of all the different cell types that we identified. We saw not huge amounts of differential methylation, but there was a decent amount. So we identified especially glutamatergic neurons that seem to have a lot of differentially methylated RNAs compared to other cell types. And this was particularly striking when we looked at the number of hypermethylated RNAs, which seemed to be particularly abundant in glutamatergic neurons versus astrocytes. And just as one example, I can show you this DDX6 transcript, which hopefully you can appreciate from these UMAP plots is fairly equally expressed in neurons versus astrocytes, but has this really striking hypermethylation in neurons specifically. So, take home is that there are many RNAs that are shared among cell types, but some RNAs can exhibit differential methylation across cell types. And we also found the same for neuronal subtypes, which in the interest of time, I won't be showing you. So one final piece of data I'll share with you before I wrap up is we've been really interested in understanding how methylation might be changing in the aged brain. So we performed single cell DART-seq in young and old cortex. Again, when we integrated the data sets, we saw good representation of the major cell types in the cortex. And when we did our differential methylation analysis in these different cell types in the young versus the aged brain, we saw some cell types that had very little differential methylation, but others like neurons and astrocytes that had a lot of differential methylation. We focused on glutamatergic neurons because they had, a really high number of differentially methylated transcripts with age. And we saw that one of the most highly differentially methylated RNAs was the APP mRNA. So this encodes amyloid precursor protein, which when abnormally proteolytically processed leads to the A beta plaques that are characteristic of Alzheimer's disease. We can see that if we look at kind of a UMAP plot of glutamatergic neurons, all of these are the cells that express APP. Again, hopefully what you can appreciate is much higher methylation in the young versus the aged brain, despite relatively similar levels of expression. We also performed a differential methylation analysis comparing glutamatergic neurons and then all the other non-neuronal cells. And we saw that this was really a neuron-specific effect. And we could validate this by performing in vitro DART-seq using bulk cortex, which again is not just neurons, but is all the different cell types. And as expected, we didn't see a difference in the amount of CDU, amount of methylation. And we could validate this at individual sites. So there are about 14 sites in APP that are differentially methylated with age. I'm showing you one in particular at site A2599, and we can look at, again, the hypermethylation seen in the young versus the old neurons. And if we validate this using an RT-qPCR-based approach, we see that, again, in bulk cortex, as we expect, we don't see a difference in the methylation amount at this particular site. Whereas when we purify neurons from mice, now we can appreciate the difference. So I think what this really highlights is the importance of single cell mapping, because this is the kind of thing that's hidden when we profile M6A from bulk cells, but now becomes apparent in single cells. So to finish, hopefully what I've convinced you of is that we can use single cell DART-seq to identify M6A in single cells, and we now have DART mice, which gives us the ability to map M6A in vivo. This has led to some important discoveries about M6A biology, namely that there are many more sites than previously thought, that these sites occur relatively rarely, and that in the brain there is relative hypomethylation in microglial cells. And also we can identify some functionally relevant RNAs and disease-associated RNAs that are differentially methylated with age. So I think with our DART-seq toolbox now, we have cellular DART-seq, which can enable single-cell M6A mapping, DART mice, which can do the same, and in vitro DART-seq, which I mentioned and didn't talk a lot about, but we're not yet there yet for single cells. And I also would be remiss if I didn't mention other antibody-based approaches, and there are other non-antibody-based approaches that are approaching single cell, which I think is very exciting. So with that, I want to again shout out to Matt Tagawski, as well as Anna Prater, who's contributed to this project, our collaborators, our funding, and I'll stop there because I know I'm already a few minutes over time.
All right. Thank you very much. And just remember, any of the participants who have questions, please feel free to put them in the chat box, which is a little bit better, or of course we can see it in the question and answer box. So let me ask a question from the question and answer box, which is, have you been able to perform viral infections or other immune challenges in the DART-seq mice, and maybe then see dynamic changes in methylation? Basically, I mean, I think this person is stimulated by the microglia question. Is it dynamic?
Yeah, this is a great question. And for those not familiar with microglial cells, they're kind of the resident immune cells of the brain. And yeah, neuroimmunological and other insults can activate these microglia. So the short answer is no, we haven't done that yet, but that's definitely in the works because we're really excited. We have this hypothesis that maybe methylation is kind of maintained at low levels in microglial cells, when they're not activated, so that there's kind of more signal to noise, right? So once you activate them, you might now get hypermethylation. And there's a little bit of evidence in the literature that might be the case. So that's a great question, but it's in the works. We haven't done it yet.
So questions are popping up. I'm going to just do Oliver Artz's question. And he's just wondering, what's the basis for the differential methylation? Is it because the writers are expressed at different levels, or why? And why is it transcript specific? Where did that happen?
Right. That's a fantastic question. In the interest of time, I didn't show the data, but we have looked at all the major writer, reader, eraser, and EJC components across the different cell types, and even with age. And nothing stood out to us in terms of, differences that might explain this differential methylation. Now, that said, that's all at the RNA level. It could be very different at the protein level. So there's that caveat. But yeah, we don't seem to think that it's due to that. So it might be a more nuanced level of something like a transcription factor, maybe influencing the recruitment of the methyltransferase to specific RNAs.
Okay, so we better switch over to our next speaker so you can unshare, and we will now switch over to Dr. Schwartz at the Weizmann Institute, who's going to talk about deciphering the code that governs methylation. So Schraga, take it away, and please unmute yourself if you're not already unmuted.
Thank you. This is good. Great. So thank you very, very much, Kate and Samie, for this invitation. Yeah, I'm very happy to talk about my work about the factors shaping the unique distribution of M6A along genes. And I'm happy I can go after Kate because it means I don't need to give a detailed introduction about M6A. I get to build on her beautiful introduction on the topic. So I'll immediately jump into the key question I want to ask here, which is basically to understand the features shaping the distribution of M6A along genes. And I want to argue that this is a fundamental question because if we want to understand different aspects about M6A, such as its biogenesis, when exactly it happens within cells, to what extent it can be dynamic and when, and how it is regulated, and how does it evolve, and how does it function, all of this is tied to a fundamental question about what governs the M6A landscapes within genes, which is something we've been struggling to understand for a long while. So going back more than a decade ago, we and also Kate published the first maps of M6A within cells. And at the time, we established this using this antibody-based approach. And we found thousands of sites within mRNAs. They're all enriched with a specific consensus motif. And the main curiosity about these sites is that they were enriched within specific regions within genes. So here are some screenshots from that original paper, where we can see what also Kate was showing with her metagene analysis. We're finding this enrichment towards the end of the genes in the region we initially characterized as the stop codon, but later turned out to be more related to the last exon within the gene. We're also finding them enriched within these long internal exons. And the question is, we've been struggling to understand is what governs this selectivity in M6A. Where is this coming from? Where is the specificity coming from of this deposition? And this is something that both Anna and Boris in my lab set out to decipher using two different approaches. So Anna, she thought she'd use a massively parallel recorder assay. The idea is let's generate thousands and thousands of mutants of different M6A sites. We can systematically mutate them, we can shift them around, and we can design short oligos, introduce them into the 3' UTR of a reporter gene, and ask which of them gets methylated in the quest for this secret component, something that causes this specificity, something we simply could not understand where it was coming from. Boris was aiming to look at somewhat longer kind of effects, so effects that have to do with introns and with reading frames and so forth. So for that, we couldn't really make use of these short oligos, and he just went for cloning of full-length genes with various mutations that he carefully introduced in order to dissect this same question, but from a slightly different lens. And due to the lack of time, I can't really show you all of these results, but the key insight we had from all of these experiments was that basically nothing seemed to be required for M6A formation. So we take a gene with introns, we remove the introns, the methylation still remains on. We can take a gene, we can disrupt the reading frame, but the methylation will still stay exactly where it was originally. We can move around the position of the methylation motif within a bunch of reporters we had used, and nothing really seemed to happen. We also tried to play around with the source of the DNA that we introduced, so whether it was from plasmids or whether we had stably integrated into cells, or whether they were linear or whether they were circular, trying in an indirect way to probe whether chromatin could somehow be responsible. And we didn't really see any difference depending on the source of DNA that we introduced. Nothing really seemed to be required for M6A to form where it originally formed. So that is where I would say, the single eureka moment in my entire scientific career. So if, whatever stage you're at and you haven't yet had a eureka moment, don't fret, maybe one day you'll have one. I just had one. And the thought was, what if there's no basis for M6A selectivity? So what if we're thinking about this entire question wrong? And instead, the only thing that's actually required is a consensus motif. And that the specificity, rather than coming from selectivity, is actually coming from exclusion of M6A from certain sites. So it's basically flipping this question on its head. And there were many reasons to believe that this would be wrong. Again, our entire mindset had been that this had to be something specific and selective because we're only seeing it at very specific sites. And yet we said, okay, let's give this theory a chance. And we built this very simple model. This is it's a model that takes 5 lines of code to implement, where the idea is, let's just take this construct over here. This is a sequence that encoding for the COM3 gene. Let's just find all of what we call eligible DRAC motifs. So these are basically a subset of 7 motifs that get highly methylated. Let's plot each of them out. And because we were relying at the time primarily on antibody-based mapping, let's just model each such site as a Gaussian. Okay, because if you were to actually enrich for such a site with an antibody, you would get a peak around it. So each site gives us a Gaussian, and then we can look at the sum of all Gaussians around all of the sites and get the predicted methylation profile that we predict for this gene. And now we can ask, okay, to what extent does this correspond to measurements? And because Boris had already generated a lot of measurements, we could just go back and relate this prediction to this measurement. So in this case, this is a measurement we have when we just introduce this gene without any intron, so only the exonic part of this gene. And when we actually look at the methylation profile that we get from using antibody-based mapping, we actually get a very similar profile to what we would predict. So in this case, it's a correlation of about 0.9. So when we look at the same measurements in the same gene, however, but when instead of encoding this gene, expressing it without the introns, we also express the introns, you can see that this entire correlation is eliminated. So for this one particular construct, it seemed that our 5-line model, was quite good in capturing profiles along single exon genes, but not when you express the same genes with the introns with them. So this model is, of course, one we can apply globally, which is what we did. And we leveraged the fact that human has both single exon genes, few of them, but also multi-exon ones. And again, we could see that within single exon genes, we actually did a pretty good job with this model of capturing the variability in M6A levels between different genes. Whereas if we look at multi-exon genes, we do a way, way poorer job in capturing this variability. So the question was, what underlies these differences between these single exon genes and multi-exonic versions and multi-exon genes? Of course, it has to do something to do. It had to have something to do with the introns, because that's the key difference between them. That's the only difference between them. So we started looking at how introns were distributed with respect how M6A sites were distributed with respect to the exon-intron junctions. We took a bunch of M6A sites mapped at single nucleotide resolution, and we just asked how do they distribute. So the very left here, the axis here is the intron-exon junction, whereas the right part here is whereas over here we're at the exon-intron junction. And what we saw is something striking. We saw that introns were highly depleted from this region of 200 nucleotides from both ends of the intron. So we're seeing that within this region here, which we call the M6A exclusion zone, we see this gradual, we see that as we're extremely close to the junction, so within a distance of, say, 10 bases from it, we have very, very few M6A sites that were ever mapped to this region. And as we extend, we see this gradual increase in the propensity to undergo methylation. And the same was true also for the other end of the gene. The same was also true when we looked at the last exon, which is also a long last exon, where again we could see the same depleted region extending up to about 200 base pairs. So with this observation, we wanted to ask whether this is a causal relationship. So we're seeing M6A being depleted, and the question is it depleted because of does the distance from a splice junction lead to inability to become methylated? And to ask this question, we turned once again to our massively parallel reporter assays. But in contrast to our very first assays, where we had used backbones that did not have any introns in them, now that we thought introns actually made a difference, we introduced introns into these, and we introduced all of our sequences into this last exon, so downstream of an intron over here. And what we did is we designed a series of sequences where we gradually shifted the relative position of the consensus motif along the sequences. So we would have ones where the sequences were very close to the introns, which if there was to be a causal relationship, we would expect these to not undergo methylation, and then there would be ones much more within which should undergo methylation. And the results we got for this experiment were as unequivocal as results can be. So you can see that as you increase the distance from the splice site, you increase methylation levels across the sites that we designed over here. So this establishes that there's a causal relationship here, that the proximity to splice junction defines the propensity and in a causal way of a site to undergo methylation. So with this observation, we could update our model, and instead of just saying that all M6A consensus motifs are modified, we're now adding this clause here, that they have to be within that they're not going to be modified if they're within a distance of 100 nucleotide of a splice junction. And we chose 100 nucleotide because it's smack in the middle of this window where we had found M6A to be depleted. So now instead of having a model which is 5 lines long, we're moving to a model which is 10 lines long. Okay, this is still a very simple model, very few free parameters here. And I'm arguing and I'm emphasizing this because I think the fact that it's so simple and that there are so few arguments is a very strong feature of this model. So we can go back to this model now, and we can ask what would the predictions of this model be now if we were to reassess its predictions now based on the same gene I had shown you before. And if we do this, all of these original sites, which originally we had predicted to undergo methylation, will no longer be predicted to undergo methylation. The reason is that they're all going to be either within 100 bases of the beginning or of the last, or of the end of an exon, and therefore too close to an intron in order to be predicted to be methylated. The only sites that would be predicted to be methylated would be the 3 here at the end within the long last exon. And this is actually, of course, typical. This structure here of having short internal exons with a long last exon is typical of mammalian exons, where that's typically the case, and where hence you might typically be able to expect this kind of a signature.
And now, I think we're not advancing the slide. I think we're still on the slide which shows the exclusion zone at 0 to 200.
That's weird.
Okay, so we're now close, but just keep in mind that we've missed some of the images that you've shown. If there's anything you want to really show in that 1 or 2 slides that we missed, just
yeah. Okay, so I'll go back. Do I get 2 extra minutes then?
Yeah, absolutely. And we can see everything moving now fine.
Okay, great. So this is where it froze.
I'm sorry, go ahead.
This is the slide where everything froze, right?
Yep, absolutely.
Okay, so just to quickly reiterate, we did this, again, we used a massively parallel reporter assay where we introduced thousands of variants, where we systematically shifted the methylation consensus motif along the length of the oligos, and then introduced them into the fourth exon here, so downstream of the intron. And the results that we got, and the idea was to check whether distance had a causal impact on M6A, and that's exactly what we observed in this experiment. So we can see that as we increase the distance from the splice site, methylation motifs become much more prone to undergo methylation, leading to this kind of linear relationship that we're seeing here, where we're looking at methylation level for each construct as a function of the distance from the splice site into which we've introduced it. And that led us to update our model to now assume that all methylation sites are going to be modified unless they're close to the splice junction, so within a distance of up to 100 bases. And when we revisited this model now, for basically for the same gene we looked at before, we're seeing that most of the sites that we had originally predicted to undergo methylation, so all of the sites here at the beginning of the gene, none of these would really be predicted to undergo methylation anymore because they're all too close to one of the two ends or both of an exon, so too close to an intron. And the only sites predicted are the ones at the very end here, leading to this very close agreement now between our measurements and our predictions for this one gene. And of course, we did this not only for this one gene, but we could now apply this model more widely. And I just kind of show you this one result. There are many more in the paper. But basically, when we're looking at predicted M6A levels, again, using this super simple model, and between the measured M6A levels, you can see that we're getting this correlation about 0.7 between the measured and the predicted levels of M6A. So that means that our model can roughly capture 50% of the variability in M6A levels between one gene and another, based only on these two simple principles that I laid out to you, that every site gets modified with the exceptions of sites close to the splice junctions. We can also kind of look at predicted versus measured profiles that we can get for individual genes. So on the top here, I'm showing you IP versus input. So this is M6A-seq-based data. These are the profiles we've been staring at and trying to understand for years and trying to dissect where they're coming from. And then below, you can see the profiles that we would get based on our predicted model. And I'm hoping you can appreciate this close agreement between the two. And if we can quantify this agreement, so on the top left, we have ones which would be the best kind of agreement. And below, the rightmost plot is one where we get 0.55, so it's less. But I think you can appreciate that along the range, and I think the mode values are around 0.6 or 0.7 for here, you can get a sense of just how well this model can actually recapitulate M6A profiles along genes. Because I'm talking in a session of where the goal is really of quantifying and mapping methylations, I also thought I'd kind of show two additional slides. One is we can, now that we have a bunch of different single nucleotide approaches to measure M6A, we could also ask to what extent does our approach actually detect M6A at the single nucleotide level. So we can look at a bunch of different approaches that provide single nucleotide resolution data, and we can ask among all of the sites that we predict to undergo methylation, what fraction of them were detected by these single nucleotide approaches versus what fraction of them were not. So for example, if you look at MyClip data here, you can see that about 30% of the sites that we detect, or 35%, were actually validated by MyClip, whereas among the ones we predict not to undergo methylation, only a very small amount of them had actually been detected by MyClip studies. So it gives a sense that overall there is a signal in our model, but there's also many sites here that were not ever detected at single nucleotide resolution, so suggesting there was also noise. But more recently, with availability of Glory, we could revisit this question, and all of a sudden we go from about 35% to about 60% of the sites being validated, again with limited, and you can see that only few sites that we predict not to undergo methylation are measured to undergo methylation by Glory. And we can take all of these methods together, kind of make our meta kind of set of M6A, of high confidence M6A plots, and you can see that we get up to about 70% of the sites that we would predict to undergo methylation are actually detected, again, with relatively low levels of noise. So sites we predict not to undergo methylation that are. We can even ask this question a different way. So under the assumption that we can use Glory as a gold standard, okay, let's assume that whatever Glory says, Glory is an oracle. And if Glory says the site is real, then it's real. And if Glory says it's not, then it's not. We can ask, what is the Jacquard index? So basically, what is the overlap between our method, between different methods and between glory. And this is what we're doing here. And we can see that using these different approaches, all of which are intense and experimental and so forth, we can get Jacquard indexes of about 0.4. And if we just plug in, again, only these 10 simple lines of code that form the essence of M6A Pred 2, and we ask, what is the Jacquard index that we get there? It actually outperforms these experimental methods in terms of the overlap. Now, I will say there are two caveats here. One, we're limiting this for expressed genes. We're also limiting this to gene bodies, and feel free to ask me about this in the question session. I want to get to one final question, which is, what is the mechanism that leads to the depletion of M6A from the proximity of splice sites? We suspect that the exon junction complex might be involved in it because the exon junction complex is this physical complex that sits upstream of the splice junctions and forms kind of this physical, so it's physically bound there and remains stably bound during export to the cytoplasm. So we thought that perhaps the presence of this junction could physically occlude the methyltransferase complex and could prevent it from potentially binding M6A sites that overlap the region bound by it. The hypothesis would be that loss of the EJC would lead to accumulation of M6A in regions where M6A is otherwise excluded. So we did precisely this experiment. We teamed up with Ervele here. He has this nice system where an auxin a degron where we can deplete Y14, which is this key component of the exon junction complex, over a 24-hour time course. And what we did is we mapped M6A along each of the time points following depletion of Y14, with the expectation that as you deplete Y14 more, you should see an accumulation of M6A in the proximity of introns in regions where we typically didn't see it. And this is actually one of those rare experiments where you come in, you have an hypothesis, you do the experiment, and it will actually fully be consistent with the hypothesis. So what we're looking here is at all M6A sites sorted based on their distance from the nearest exon junction. So basically based on proximity to introns. And you can see that sites here in close proximity are basically not methylated to begin with, but strongly accumulate methylation over the course of this depletion. Whereas if you look at sites that were more distant to begin with, say sites over 200 bases long, you can see that basically they change to a very little extent over the course of this time course. So consistent with the hypothesis that the inability of these sites to become modified in the proximity to the splice junction is due to the exon junction complex. So this leads us to a very simple model, and there are some. We can think of all sorts of variations of the model, but the simplest model for that we came across with is that that within is basically one of physical occlusion. So that within short exons, you will have the exon junction complex binding. You will have the short exon junction complex binding there, and the exon junction complex, perhaps in conjunction with different RBPs, which are recruited by this complex, simply prevent the methylation complex. This methylation complex from being able to access the area and to methylate RNA. Whereas if you're looking at longer internal exons, you don't have this region being sequestered, allowing M6A to be deposited on the RNA. I'd like to highlight here 3 additional papers who came to very similar conclusions recently by Richard Gregory's lab, by Xuan He's lab, and by Shengdong's Ke lab more recently, so about a week or 2 ago, all of which kind of converging in similar conclusions on this topic of the exon junction complex. Okay, so just to briefly summarize and speculate perhaps. So I guess the key conclusion here that M6A deposition is highly predictable via a simple 100 base pair rule. Now, of course, the model is oversimplified. We're currently working on a much more quantitative model guided by much more quantitative measurements that we have available at the moment. With this, we'll be able to actually plug in numbers, perhaps assign weights to different consensus motifs, and be able to better model the distance from the splice site rather than taking a fixed cutoff. Perhaps we'll be able to take to use this as a more continuous kind of variable, but it won't change the fundamental aspects of this model, which allow us quite a high ability to predict M6A levels on the basis of this very simple rule. Consistent also with the point Kate was making about finding way more sites, this is also what our model converges on, right? So we can use this model now to ask how many M6A sites are there, and we converge actually in quite similar models, so around 200,000 sites with an average 12 sites per genes. And these are actually also similar numbers to what glory measurements are converging on. So I think all of us were misled by studies originating, for which I take responsibility for, in part, where perhaps inspired by some of the recent literatures from the 1970s, where the assumptions were there were going to be 2 or 3 sites that were going to be modified at high stoichiometries per gene. We're all kind of thinking along these lines. And I think now with better measurements and better ability to understand where M6A is coming from, I think the understanding is there are way more sites per gene, but at way lower stoichiometries than perhaps we had initially thought. And I think this has important implications. I think this and also the findings on how this is tied to exon intron architecture has important implications on M6A regulation and dynamics. I think with the understanding of what shapes the M6A landscapes, it gives us an ability to think about what kind of parameters might alter how M6A is distributed. So for example, if M6A is tied to splicing, then changes in splicing, one could envision how they might tie to changes downstream in M6A levels. I think the far largest number of sites that we're talking about here, if we think about 12 sites per gene, I think it also kind of causes us perhaps to reconsider what the relevant resolution even is for quantifying M6A. And I think this might be an interesting topic for the panel later. I think a lot of the efforts have been concentrating on quantifying M6A levels at individual sites, and then perhaps trying to connect them to phenotype. But I've been thinking for a while that perhaps what we need to come up with is much better metrics for quantifying the overall M6A load per gene, right? And how do we integrate over different numbers of M6A along a gene, and to use that metric at the gene level to then connect M6A load to functions and to downstream consequences. And finally, I'd like to highlight that there's this, with M6A being attached early on in the nucleus, downstream of an early processing event, splicing, and then in turn connecting to cytoplasmic degradation, it kind of imposes M6A as this integrator of signal between nucleus and between cytoplasm. And with all of the evidence for different levels of coupling between the nucleus and the cytoplasm, it's exciting, I think, to consider the roles that M6A might be having in shaping these connections, given how it's deeply tied and mechanistically tied to both. So I'll end with here. I'm happy to take any questions. Once again, I'd like to highlight the people doing this research. So wonderful PhD student Anna Ozoni, the great Boris Lobonin, who's meanwhile opened his lab at the Technion. He was a super talented research associate. And this was done in collaboration with Hervé Lehir. He's the guy who actually discovered the exon junction complex and happened to spend time in Israel as we were thinking about all this. So it evolved into this beautiful collaboration. And I'm happy to take questions. Thank you.
All right. Wonderful. You know, we're running a little bit late on time for our break, but I just want to say, I think during the panel session, one of the things we're going to talk a little bit about is some of the very important methods you've developed, some of which were used here, like the M6A-seq2, and when M6A-seq2 should be used, as well as master-seq, and what's the role of master-seq, which is a method that we'll briefly touch on in the panel session, what is its current role. But I think another question directly related to your talk that will probably come up towards the end of the panel session is, how do we integrate your story with another story in Nature that says SETD2 modifies histones, and then another story that says Verma recruits the methyl transferase complex to the terminal exon junction complexes? How do we how do we integrate all these different models? Are they all working together? Is the EJC model the primary model? And what about those other models? And then I think another question is, what does your model say about dynamics of M6A? Because your model really argues that genomic architecture alone, maybe I shouldn't put words in your mouth, but genomic architecture has a big effect on methylation. But, I'll let Schraga address these questions in the panel.
Are you seeing my slides?
Yes, we are. But we see it with the slide. Now it's right.
Advances?
Yep.
Great. All right. I want to start by thanking Samie and Kate for organizing this virtual symposium. I thought this is a great opportunity for us to gather and discuss the newer methods and technologies in the epitranscriptomic field. And I'm pleased to be a part of this. In the next 20 minutes, I'm going to tell you a bit of our story. So we developed a method for transcriptome-wide profiling and quantification of N6 methyladenosine, and our method is based on enzyme-assisted global adenosine deamination. I think with the first 3 talks, I no longer need to prep the audience on this particular modification, N6 methyladenosine or M6A. This is the most abundant internal mRNA modification in higher eukaryotes. It participates in virtually all aspects of the mRNA life cycle. It has rich biology. In order to understand that biology, we first need to know where the modifications are. About 10 years ago, 2 state-of-the-art M6A mapping methods were developed. M6A-seq or ME-RIP-seq, both function by immunoprecipitation, basically harvest the RNA, fragment them, use the antibody that recognize M6A containing transcripts and enrich them. You sequence the input pool, you sequence the enriched pool. Where you see the enrichment would indicate the presence of M6A. These 2 are very elegantly designed methods, very robust. A lot of what we know nowadays about M6A is derived from these methods. However, they do have some inherent limitations. For example, if you look at the enrichment peaks, they're typically a few hundred nucleotide wide. It would be harder for us to know where exactly M6A are, or how many M6A sites you have in that one peak, or to what extent a particular site is methylated. On the other hand, in order to support the immunoprecipitation workflow, we tend to need bulk input materials. We set out to develop a method that has orthogonal detection principles to address some of those limitations. Before I introduce you our method, I would like to briefly touch upon similarly bisulfite sequencing, the classic method that reveals the methylation landscape in DNA. So this is also the inspiration of our method. In bisulfite sequencing, cytosine is deaminated or hydrolyzed into uracil, and this reaction will not occur on 5-methylcytosine. So basically, during bisulfite treatment, you degenerate the entire genome into 3 codes, T, A, G, and the remaining C will tell you where methylated C is. Using this short piece of sequence as an example, this sequence has 6 Cs. 4 of them are unmethylated, 2 of them are methylated. If we directly sequence this particular DNA, we would lose methylation information because all of them will show up as C. However, in bisulfite sequencing, the unmodified C will show up as T, and the persistent C will tell us, okay, it is these 2 Cs that are methylated. With bisulfite sequencing being there for decades, it's perhaps not too difficult to come up with this hypothetical method for M6A detection. What if we can convert all unmethylated A into inosine without impacting M6A? Then we would be able to degenerate the entire transcriptome into 3-letter GCT. And the remaining A will tell us where M6A is. By the way, inosine will be read as G by reverse transcriptases, polymerases, etc. So what's the challenge? The idea is simple. In fact, A is inherently more challenging to deaminate. Let's say that I throw a single-stranded DNA into water. It's neutral pH. I didn't supply any catalyst. We would readily see C deamination. However, A deamination would occur almost 100 times slower. So this is we're looking at a more, I would say, substrate that's more chemically resistant to deamination. So at the time, we thought, okay, what about to do it enzymatically? Of course, now with Chengqi's talk, we know that it's possible to come up with small molecule catalysts to accelerate this reaction as well. But we decided at an early point that we want to pursue an enzymatic route. We screened a pool of deaminases, including hyperactive adenosine deaminases developed for gene editing purposes, and found one. This enzyme is called TAD-A 8.20. This is a laboratory-evolved TAD-A. TAD-A is a E. coli tRNA adenosine deaminase. TAD-A 8.20 would deaminate A efficiently into inosine globally without impacting M6A. So TAT-A 8.20 is called TAT-A 8.20 because it went through 8 rounds of directed evolution and happened to be the 20th candidate characterized. Okay, so now we have an enzyme that can support this workflow. In fact, with the workflow showing here, I would like to highlight 2 key challenges associated with this detection principle, which I call detection by subtraction. These 2 key challenges are efficiency and selectivity. If you look at this deamination reaction, if this is a normal organic reaction, we would consider 80%, 90% yield very good. However, 80%, 90% yield would mean that 10% or 20% false positive signals, which will overwhelm M6A detection. In that sense, TAT-A 8.20 is efficient. The second key challenge is selectivity, whether the proposed reaction would fully differentiate A and M6A. In that sense, the selectivity of TAT-A 8.20 is perfect, means that if a particular A is methylated, TAT-A 8.20 will never ever act on it, no matter how long you incubate, how much enzyme you add. To show you how the enzyme behave, I'm going to use this short RNA, synthetic RNA probe that is 53 nucleotide long. It contains about 12 unmodified A's here. I'm showing you sequencing results post-deamination. So here, A is in orange, G is in blue. You can see all these A's are detected as G, means that our enzymatic treatment is potent. I'm highlighting this particular A because at this position, I will start to dose in M6A. When I dose in 25% M6A, I see about 25% persistent A signals. When I dose in 50% M6A, I see about 50% persistent A signals. When this position is fully occupied by M6A, I detect only A signals. So with these findings, we can get to 2 conclusions. 1, the enzymatic treatment is both potent and selective. 2, it is possible for us to detect M6A quantitatively by comparing the signal intensity of A and G post-deamination at a given site. So with that, we incorporated this enzymatic treatment into classic RNA-seq workflow and developed this method we called evolved TAT-A assisted N6-methyladenosine sequencing, or ETAM-seq. We applied ETAM-seq to HeLa cells. We were able to detect the 2 known M6A sites in human ribosomal RNA. Both sites are methylated close to completion. In the HeLa mRNA pool, we detected about 70K M6A sites methylated to greater than 10%. Our results are reproducible because the methylation level estimated using 2 independent biological replicates agree very well with each other. Most of the detected sites are found in DRAZH motif, the motif known to host M6A in higher eukaryotes. Similar to what observed with Glory, we also saw a considerable portion of these M6A sites are actually methylated to high levels. For example, here, 20K of the detected 70K M6A sites are actually methylated to greater than 70%. I would like to note here that the 70K sites that we detected in HeLa transcriptome is not the upper limit of human transcriptome. In fact, this number is highly impacted by sequencing depth. If you don't sequence enough, you tend to lose 2 types of sites. One type is lowly methylated sites, for which you need to sample more times in order to confidently say that, okay, I have methylation signal there. The second type of signals you tend to lose with inadequate sequencing depths would be M6A sites in lowly expressed transcripts. So by no means this is the upper limit, and we actually know that we haven't saturated detection yet. I would also note that we use the control transcriptomes prepared by in vitro transcription or FTO treatment to get rid of some of the false positives. All right, how would the ETAM-seq look like when we inspect individual genes? Here I'm giving you a couple of examples. The top track is immunoprecipitation data, ME-RIP. Here input is in pink, IP is in blue. So wherever you see a larger blue peak means that there's enrichment and hence M6A. The bottom track is ETAM-seq data. ETAM-seq is base resolution, so every peak corresponds to 1 M6A site. The y-axis reflects the methylation level. For example, if you look at this site has this peak is high, means that the methylation level here is high. Some of the other peaks are not as high or lower, means that those sites are methylated to a moderate or lower level. When we plot data in this way, one thing that becomes immediate apparent is that most of the ETEM-seq peaks locate within or close to immunoprecipitation peaks, means that these two methods with orthogonal detection principles actually agree well with each other. Another, perhaps not too surprising, my slides are not advancing. Okay. Another perhaps not surprising observation is that one immunoprecipitation peak typically covers more than one M6A sites. I think this has been hypothesized for a while, and we are offering a piece of evidence to support that hypothesis. Similar to what we heard in the previous two talks, one gene also tend to contain more than one M6A site. We saw a tendency for M6A sites to cluster, which makes it tempting to, correlate M6 or do an analogy of M6A distribution to the methylation island we observed in DNA. I think it's a very reasonable hypothesis to assume that clustered M6A may have stronger function, but I don't think we have enough evidence to support that. All right, I would like to highlight one feature associated with ETEM-seq that had a 8.20 treatment is mild. We already know that bisulfite treatment tend to degrade DNA. RNA tend to be more sensitive to degradation. So a natural question here, before we even do the RNA-seq, would be to take a look at the degradation. How would the mRNA behave when we incubate them with the enzyme? So we analyzed the RNA treated with or without the enzyme, and we saw that the size of the fragmented mRNA didn't change, means that we saw minimal degradation. With minimal degradation, we were able to start with fewer starting materials. Throughout the initial study, we used 50 nanogram of mRNA. This is actually not the detection limit. We didn't try to probe the lower detection limit. I got asked by some researchers whether we tried lower than 15 nanogram. We actually didn't try, but I would confidently say that we are far away from the detection limit. On the right, I'm showing you a piece of more recent data we got in lab. Here, we processed the full-length mRNA or total RNA extracted from HeLa cells under similar conditions and analyzed the RNA on a bioanalyzer. So here, these are post-deamination full-length mRNA. You can see that the average length is approximately 1,000 nucleotide. This is shorter than the expected average length of human mRNA, which I believe should be 2.5K. However, we think that this level of degradation is manageable. And in fact, within this post-deamination pool, we should still have plenty of intact mRNA molecules that would support us to do single molecule end-to-end M6A profiling. I thought that would be a very exciting direction, and we are actively pursuing that. So now moving away from HeLa, we also applied ETEM-seq to mouse embryonic stem cells, mouse ES cells. A lot of what we observed with HeLa was actually recapitulated in mouse cells. For example, we detected 60K M6A sites of greater than 10% methylation. A considerable portion of them are methylated to a high level. Our hits are enriched in DRACH motif. Results are reproducible across biological replicates, and our results correlate well with existing methods, especially immunoprecipitation. I'm not going to bore you by showing you similar type of plots for mouse cells, but I would like to share a specific experiment we did with mouse ES cells, which I think will give you a sense of the fidelity of our detection. We have engineered a mouse ES cell line in which we can conditionally knock out METhyl3, the methyltransferase, the core methyltransferase responsible for M6A deposition. We can apply ETEM-seq to both the control mouse ES cell and METhyl3 knockout mouse ES cells and compare the detection. I thought that would give us a good idea of the fidelity of the method. So satisfyingly, upon depletion of METhyl3, we detected a much fewer M6A sites. In fact, in the control mouse ES cells, we detected about 10K cells. Again, the number of M6A sites you detect are highly correlated with sequencing depth. Here, we didn't sequence as deeply. And in the MELO3 knockout strain, we detected about 2.7K M6A sites, which means upon MELO3 depletion, 70%, more than 70% of M6A sites disappeared. If we further inspect the methylation level within those detected sites, in the control cells, the average methylation level is slightly over 50%. And in the MELO3 knockout cells, the average methylation level is about 20%. If we look at the 2K sites that overlap between the control cells and knockout cells and plot their methylation levels in the control cell line and knockout cell line this way, you'll see that most of the dots enriched to the bottom right means that they have a higher methylation level in the control cell line compared to MELO3 knockout cell line. Bottom right just showing you a distribution of methylation level changes. Collectively, we show that both the number of M6A sites and the methylation levels decrease upon MELO3 depletion. I thought that also offered a good piece of evidence to support the fidelity of our detection. In addition to transcriptome-wide profiling, we are aware of the potential application of site-specific detection. Some researchers may only care about a few sites, whether they're methylated. If so, to what extent they're methylated. It would be cost inefficient if we need to extract that information by RNA-seq. So motivated by those applications, potential applications, we developed a workflow that we call ETEM-Sanger. This is very similar to ETEM-seq. You still harvest your mRNA or total RNA, you fragment RNA, you deaminate, you do reverse transcription. At this step, instead of building an RNA-seq library, I would design a site-specific primer to amplify the site of interest. Of course, your primer will have to recognize post-deamination sequences instead of the original sequences. And you can sequence that amplicon either by Sanger sequencing or amplicon deep sequencing. We attempted this workflow on about 20 sites. Here I'm showing you 8 examples. Those are Sanger sequencing data in which G is in black and A is in green. I already highlighted the putative M6A sites, but I would like to first draw your attention to some of the surrounding A's. Those surrounding A's, since they're not methylated, they would be susceptible to enzymatic deamination, so they are detected as G. At the putative M6A sites, you will start to see persistent A signals. And by comparing the intensity of A versus G, we would get to a quantification value, how much methylation you have there. Some of the sites are methylated close to completion, for example, this one. Other sites, you will see a fraction of G signals that would correspond to here, maybe 70% methylation. We hope that with this workflow, researchers would be able to routinely survey M6A levels in their biological processes of interest. And since the enzymatic workflow is mild, we were able to demonstrate M6A detection and quantification as few as 10 cells, in other words, 250 picogram of total RNA. In the last slide, I actually want to share a piece of ongoing work in my lab. Since the publication of our initial story, we carried out additional directed evolution in my lab. We isolated an enzyme that we named TADA8R, which is more active and context compatible than TADA8.20. In our hands, in vitro, TADA8R is about 10 times faster than TADA8.20. I'm showing you data on DNA, but we have similar data on RNA. By integrating TADA8R into ETEM-seq, we hope to further improve the sensitivity and fidelity of our method, and I hope to report those results to the field in the near future. With that, I would conclude. I want to thank my lab, especially Yulan Xiao, a postdoc, and Yuan Wu, a graduate student, who bravely took the project knowing that their advisor knows little about M6A and genomics. And I also want to thank my collaborators, Dr. Chuan He and Dr. Meng Jiechen, who generously offered their expertise. Their help was instrumental through every step of this project. I don't think the project would be there without them. I want to thank Funding Resources, NHGRI, and Source Scholars Program. And thank you for your attention.
Weixin, thank you very much. Just a really fascinating method. And I think in the panel discussion, we're going to talk more about ETAM-seq versus Glory. I don't want to use the word versus. I'm not saying that they're fighting each other, but I think it's really going to be a lot of discussion about which one to use under what contexts. And then even Dart-seq, when should we use that technique, and even Master-seq and M6A-seq, if there's any future for M6A-seq / MIRIP-seq that we developed many years ago, if that has any value at all. But let me, people can ask questions, but let me ask a question about this enzyme. Is the TAD 8.20 commercially available? And if you make it, how stable is it? Like, is it a stable enzyme? Is it easy to work with?
Well, thank you for bringing that important question. Unfortunately, it's not commercially available. I hope some vendors, I don't know if there are vendors in the audience. If you're interested, you should totally reach out to us. The enzyme itself is quite stable. However, we found that the enzyme doesn't express well without a solubility tag. Means that you will have to express it with MVP, and at a later stage, you will have to remove it, which will make the purification process longer than usual. We purify other enzymes in our labs. So it wasn't a super challenging task for us, but I realized that not every lab has expertise to do protein purification. So I would put out an advertisement here, in fact. So if you're interested in our method, please reach out to me. I will send you an aliquot of enzyme enough for 20 reactions. So I welcome you to try out before you commit the effort to protein prep. We have done this to now dozens of labs. I don't expect any credit from your work. Just reach out to me.
Wonderful. Let me ask, Claudia Colina Prisco asks, how do you incorporate endogenous ADAR editing, the A to I editing, and how does your algorithms to map sites take that into account, or does it matter?
ADAR would. We did a comparison. First of all, ADAR would not give you false positive signal because it's deaminating A into G. So in that sense, it's not a huge concern already. On the other aspect, we frequently utilize in vitro transcribed transcriptome or FTO treatment to serve as a controls. ADAR sites will show up in those control samples as well, so we would be able to readily get rid of them. Okay. The short answer is that they don't really overlap, in fact.
Yeah, very good. But the A to I, if you are looking at, if you're comparing no enzyme to enzyme, then the A to I would be the same in both samples. But so are you comparing no enzyme to enzyme to TAD 8.20, or are you just counting the ratio of A's to I's at every individual site after enzyme treatment?
Not sure if I fully understood that.
Is your control no enzyme treatment?
When we analyze ADAR sites, we used other people's data, RNA-seq. So there's no enzymatic treatment. But in our workflow, the control transcriptomes are also treated.
Okay, got it. The Ankit Jana asks, how do you figure out the amount of enzyme? Does it change depending on the context or the amount of RNA that you add or the cell type?
So in our approach, enzyme is actually overdosed. It's not a catalytic process. The hypothesis here is that your mRNA, your RNA is more precious. So if you calculate the ratio of enzyme to RNA, it's always more enzyme. And we know that within that range, if you lower it by a little bit or increase it by a little bit, it's not going to make a difference. So it's already within a stable range that will give you enough or high efficiency deamination, and you don't have to adjust with the RNA amount.
Okay. And maybe the last question before we go to the panel discussion, and during the panel we may ask some of the other questions, is, Veronica de Jesus asks, could this be done in cells? Could you express the TAD enzyme? And, I think they're inspired by Kate's approach. Can you, induce it and then map the sites that way?
I think that's a valid idea. Comparing to DART-seq, I think we are unlucky that deamination is also on A. So how would you, and we don't have an AA motif that can, be used to back deduce where M6A is. So I think data analysis would be more complicated. But we know that this enzyme is more active than APOBAC on RNA, so which may be valuable. You will have higher signal levels, but bear in mind that data deconvolution is not going to be as straightforward.
Yeah, and I think I had a little brain freeze on the ADAR question. Now I realize that even if some adenosines have not been deaminated by ADAR, the TAD 8.20 will get the rest of them, and they'll all be deaminated to inosine and be read as G. All right. So with that, we will convene our panel session, and it looks like we're going to start our panel session right now. I think we don't have a break, so Weixin, no break for you, but we're going to pull everybody back up